Okay, good morning. So today I'm going to talk to you about the athlete's biological passport and how our improved knowledge of longitudinal biomarker variability has revolutionised the fight against doping. So I'll begin by focusing on blood doping, explain what it is and how our knowledge of blood marker variability has improved the fight against doping. I'll then explain what the biological passport is, and then go into more detail about how the passport calculates biomarker variability, and discuss one project that I did last year focused on within subject hemoglobin variation. I'll then discuss the current limitations of the passport, and that is that the majority of the blood markers that the passport uses are concentration-based and are thus influenced by plasma volume variations. I'll then discuss the future directions of the passport and how this idea of longitudinal biomarker monitoring can help in the clinical world. So when we talk about blood doping, we're obviously talking about improving our ability to transport oxygen to the working muscles. So we're mainly focusing on endurance sports here. So there are a number of factors that will improve an athlete's aerobic capacity, and this is hemoglobin mass. Hemoglobin is a blood marker which carries oxygen on the red blood cells. Then our efficiency to transport oxygenated blood from the heart to the muscles, and then how efficient we are at extracting this oxygen to the muscles. Cardiac output and oxygen extraction are relatively fixed within a human, and so the factor that's open to manipulation is our hemoglobin levels. And so this is the target that blood dopers aim to improve. So in order to catch the dopers, we must know what the aims of the dopers are, and this is to improve the levels of red blood cells in the body. And this can be done in two different ways through the pathway of red cell production. So erythropoietin, or EPO, is a hormone that stimulates red cell production from the red bone marrow. The first way to manipulate this pathway is to introduce an artificial erythropoiesis stimulant. This will in turn induce the bone marrow to produce more red blood cells and thus increase the oxygen carrying capacity of the blood. The problem with this technique is that the molecules used to stimulate red cell production are difficult to detect, and especially with the use of microdosing techniques. The second way to improve our oxygen carrying capacity is to directly increase the number of red cells in the body, and this can be done through blood transfusions. The difficulty with this technique is that you're introducing your own red blood cells, so there is no foreign material that can be detected. However, both these techniques result in changes in a number of blood cell parameters, including hemoglobin, hematocrit, our red blood cells, and our reticulocytes, which are the young red cells. And so this is what the athlete's biological passport aims to identify, any abnormal fluctuations in these markers away from the norm. And we've seen that these blood doping techniques have tainted sports. We have Johannes Doer was tested positive for the blood booster EPO just a few months ago at Sochi. And we cannot talk about blood doping without talking about cycling. Here is a table of the top 10 place getters at the Tour de France since 1998. The boxes with a face in them or darkened out are athletes who have either been caught doping or have claimed to dope in the past. So we can see this is a big problem in sports today. So how can we identify the dopers? Well, to enhance our athletic performance, there must be some sort of physiological change, and this change must have an origin. Originally, anti-doping technologies have used direct detection techniques where they directly detect the banned substance within the athlete themselves. However, we've moved to a more indirect detection technique where we're focusing on the physiological changes that result from these doping techniques. And this is where the athlete's biological passport sits. So the idea behind this system is that the human physiology has taken years and millions of years to evolve. And thus the human specimens we are today are a fairly stable parameter. And these highly suspicious performances that we have seen in the past cannot be naturally occurring. So this is the athlete's biological passport. It follows a number of, it uses a number of markers, reticulocytes, hemoglobin, hematocrit, red blood cells, and so forth, and is based on the idea that these blood parameters are relatively stable within the human body. As an athlete will give a blood sample to the anti-doping agencies and begin to develop a biological passport, which looks something like this. As the athlete introduces more blood markers or gives more blood samples to the passport system, they will begin to develop their own reference range. The reference range is shown here in the red lines, and the athlete's own actual values are shown here in the blue. So the passport follows individual athlete values over time and to develop these individualized reference ranges. So an athlete will have a blood collection. This will be analysed by WADA-accredited labs. WADA is the World Anti-Doping Agency. And these results will then be introduced to an internationally accessible electronic record, which will then be analysed by the experts. And these experts will look at these profiles and also look at what this athlete has been doing in the past. For instance, if they've been at altitude, this will also influence the athlete's values, but is a legal means to do so. And we've seen that the passport system has had great success. We've seen a great reduction in the number of abnormal values since its introduction in 2008. So we know the system works. So I'll now talk about how the passport calculates these individualized reference ranges over time. When an athlete introduces their first blood sample to the passport, it will be this value will be compared against a population-based reference range, shown here with the red dots. I will use hemoglobin concentration as an example here. From this value, the passport then uses statistics referred to as Bayesian statistics to predict what reference range this individual athlete should have. And as the athlete collects more and more blood values, this reference range will become more specific to this individual athlete. What the passport aims to identify is any abnormal fluctuations away from these normal natural variations in hemoglobin. And this may be questioned and analysed by the experts. What is important to understand is that after 3 values have been collected, this reference range calculation relies heavily on within subject hemoglobin variation. That is, this is a calculation of how much our hemoglobin can vary naturally. However, this value used within the passport today is based on a cohort of only 124 male athletes of various sporting disciplines. And so this brings me to my first study, which I did in collaboration with the Australian Institute of Sport, Aspetar, and the University of Canberra, which was focused on verifying this current estimation for within subject hemoglobin variation. So the aims of this study were to identify the athlete's biological passport value for within subject hemoglobin variation and investigate whether analysis system, sex, sport, or season influence this value in any way. So I started up with a database containing over 22,000 records collected at the Australian Institute of Sport since 1997. And after a quite extensive data cleaning process, I ended up with a database containing just over 13,000 records from healthy male and female athletes aged 15 to 36 years with a minimum of 2 hemoglobin values each. So we had a solid platform to work upon. We had over 13,000 hemoglobin records from 2,449 athletes. We had over 6,000 female observations and over 7,000 male observations, all of which came from 39 different sports. So what we did was we created a model to calculate within subject hemoglobin variation. We did this using linear mixed modelling techniques, using the R function. And our aim was to quantify within subject hemoglobin variance. So for the statisticians out there, our response variable was hemoglobin. Our fixed effects were analysis system, sex, sport, and season, and our random effects was the athlete. The first assumption that we had to meet in this modelling process was that the hemoglobin values used were independent of each other. And so our first step was to create a model that accounted for this within subject autocorrelation. So what is autocorrelation? It sounds like a pretty complicated statistical term, but it's easy to understand if we look at dress codes in the world. If we look at a country such as Qatar, where everyone lives quite close together, we see that the people dress in quite a similar way. But if we travel far enough away, we find that the people will dress very differently. This is autocorrelation. The Qataris are correlated to each other. They dress quite similarly. So are the Australians. They dress in quite a similar manner. But the Qataris and the Australians are independent of each other. So what we asked then was, how far do we have to travel for two hemoglobin values to be independent of each other? And with the model that we came up with, we found that after 35 days, if two hemoglobin values are taken 35 days apart, they are independent of each other. And this is extremely important for the passport system, as if we have values taken less than 35 days apart, they will be correlated, and this will influence the calculations used within the biological passport. So the next step, we then went on to look if analysis system, sex, sport, or season had any influence on the variation in hemoglobin. We first looked at analysis system, and we found that all the different analyzers used were quite similar, and also that our estimation for within subject hemoglobin variation was quite similar to the one currently used within the passport. We then went on to look at sex. We know that females have lower hemoglobin values than males, and this is due to blood loss during menses and also lower energy intakes. Currently, the majority of the literature is focused on male athletes. So our question was, is within subject hemoglobin variation greater in females? And what our results found was that no, in fact they are not. And this is an extremely important finding, as it justifies the use for a universal value for hemoglobin variation, which is currently calculated off male athletes and used for female athletes. So we've justified this. We then went on to look at the different sports. What we found was that there was no significant difference away from this universal value currently used within the passport. However, we found that endurance and team sports were more variable than the other sporting groups. And we predicted the reason for this was most probably because endurance and team sports experience greater shifts in their plasma volume. And as hemoglobin is a concentration-based value, it is most likely the reason for this increase in variability. When we then went on to look at season, we found that hemoglobin was lower during the hot months in comparison to the cold months, further confirming this assumption that is influenced greatly by plasma volume shifts. And we also found that during the hot months there was more variability in the hemoglobin measures than in comparison to the cold months. So the conclusions that I made from this study was that hemoglobin values must be taken more than 35 days apart. There was no differences between sex, so a value based and calculated on males can also be used for females. We found differences between different sporting groups and seasons, but this is most probably due to shifts in plasma volume. And we justified the current estimation for hemoglobin within subject hemoglobin variation in athletes. So where can we go from here? So at the moment, we're using a universal value for within subject hemoglobin variance. But we know that each individual varies in a slightly different manner to each other. So what if we introduce not a universal value to the passport, but an individual within subject hemoglobin value? This is potential to improve the specificity of the current system, but further research is required. I'll now talk about the major shortfall of the biological passport, which is that the majority of the blood markers used are concentration-based, and this means that they are influenced by shifts in plasma volume. Plasma volume is our body water. Plasma volume is influenced by the time of day, the temperature, and also influenced by exercise. And so this brings me to my next study, which I'm currently doing here at Aspetar, where the derivation of a marker for plasma volume shifts from high throughput biological data. What we've done in this study is we aim to create a panel of biomarkers that are able to correct blood data for the natural variations we see in plasma volume. And we want to introduce this model into the biological passport to make it more specific. So what we've done is we've collected blood values and also calculated plasma volume from a cohort of 33 male endurance athletes over a 6-month period, where every month we are collecting values from the athlete. And we aim to link the values that we find to try to create a panel of biomarkers that accounts for these shifts in plasma volume. The results are yet to come. We hope that this study will improve the specificity of the biological passport, but also help in our knowledge of volume management in critical care patients, and also improve our understanding of exercise and heat-triggered changes in the organism. So I've been talking a lot about the athlete's biological passport, and more specifically the blood module of this passport. But recently this year, the WADA has also introduced a steroidal module to the biological passport, which focuses on fluctuations in our hormones. A next step from here could possibly be that we link these two models together. So changes that we see in hormones may correspond to changes we see in blood measures, and this would give us a more whole body approach to catching the dopers. So where we can also go from here is to look more deep. We've been looking at the phenotype, which are the blood markers. But what if we look below this? What if we look at the metabolome, the proteome, or the genome, and calculate patterns of fluctuations in these markers to identify doping cases or potentially disease? Our knowledge of the genome is not so great at the moment, but there's been great advances in metabolomic analysis at the moment. So a metabolomic profile will look something like this. It basically looks at the charge to weight ratio of a compound, and we're able to identify over 20,000 different metabolites using this mass spectrometry analysis. So we hope that we can use these new technologies in the studies of pathologies and create specific markers for different disease states and potentially to identify doping cases. And use the same idea behind the biological passport, which is looking at or calculating individual reference ranges, but rather using these metabolites to calculate these reference ranges. We can use prostate cancer as an example for this. At the moment, we use, to identify prostate cancer, we use the prostate-specific antigen, PSA. 80 million tests are done per year, and of these, 12 million are positive. The problem with this test is that it does not differentiate between prostate cancer and benign hypertrophy. And so the test, to identify prostate cancer, we must do a follow-up biopsy. 75% of these are negative. So this is a highly invasive technique, and it is questioned as to whether this is an efficient technique. However, if we use metabolomics and metabolomic analysis, we are able to differentiate between this prostate cancer and benign hypertrophy. So both the tests are highly sensitive, that is, they're able to identify the true negatives compared to a positive. However, the current PSA test is not highly specific. It cannot differentiate between these two conditions. But if we look at metabolomic analysis, we can easily differentiate, easily identify prostate cancer using very specific metabolomic markers. And so this has great potential in the clinical world. What if, so we could use metabolomic markers to identify different pattern or individual reference ranges in health. We say that it should take 6 weeks of immobilization to heal a broken bone, but is this 6 weeks for everybody? What if we use the concept of developing reference ranges using biological markers to predict how quickly an individual will recover from such an injury? We could also use this to look at the recovery of hemoglobin after blood donation. We suggest 20 to 60 days, but what if some individuals recover more quickly than others? We can, again, what if we can find recovery markers using analysis techniques such as metabolomics to create a biological profile for individuals? Okay, so in conclusion, the athlete's biological passport is an indirect approach and has played a significant role in doping today. It is based on calculating individual reference ranges for athletes using within subject variability calculations and has been highly successful. However, plasma volume is one current limitation of this profile, of this system at the moment, and work must be done to improve this. The concept of individualized reference ranges has also great potential in public health today as well. Yeah. Thank you very much.